This graph represents a global crisis in motion. By 2050, researchers estimate antibiotic-resistant infections will cause 10 million deaths every single year. That projection is rooted in current data. In 2019 alone, 1.27 million deaths were directly caused by these resistant infections. To put that in perspective, antimicrobial resistance is already killing more people globally than HIV/AIDS or malaria. Humanity first began mass-producing the tools to fight these infections in the mid-20th century. At a microscopic level, antibiotics function as highly specialized chemical weapons. These chemicals are engineered to identify and disable vital bacterial structures, like the machinery building the cell wall or the ribosomes maintaining cellular integrity. For decades, we have relied on these drugs to make surgeries, injuries, and routine infections survivable. But that historical reliability is fading. The declining efficacy of our drugs is a predictable biological consequence of humanity's interference in the Darwinian arms race. Bacteria reproduce with extreme efficiency, often multiplying from a single cell into a population of billions within hours. Because this replication happens so quickly, it is often imperfect. This results in constant random genetic mutations appearing throughout the population. When we introduce an antibiotic into this environment, we are applying a severe environmental stressor. Most of the bacteria are susceptible to the drug and are rapidly wiped out by the chemical attack. However, out of those billions of cells, a single bacterium might possess a random, lucky mutation that allows it to survive the chemical agent. This lone survivor then replicates without competition, quickly filling the void with a new population that inherits that exact genetic immunity. Antibiotics do not create resistance, they act as a filter. They kill the vulnerable and ensure that only the most highly defended mutants survive to populate the environment. This evolutionary process has resulted in a suite of sophisticated, genetically encoded biochemical defense systems. The most effective masters of these defenses are the escape pathogens, a group of 6 species designated as critical threats to global health. We see these defenses in action at the molecular level. Mechanism 1 shows enzymatic inactivation. Bacteria produce enzymes like beta-lactamases, acting as a molecular security force. They identify incoming antibiotics and chemically dismantle them before the drug reaches its target. This specific countermeasure is what has made penicillins ineffective against a wide range of modern infections. Pathogens have evolved a way to disarm our pharmaceuticals on contact. If a drug breaches the outer membrane, it hits a second line of defense. Mechanism 2 shows an efflux pump. These function like biological bailing systems, physically grabbing antibiotic molecules and forcing them back out of the cell. These pumps are often generalized, meaning they can extrude several different classes of antibiotics, giving the bacterium multi-drug resistance in one move. The final internal defense is the alteration of the drug's target, illustrated at the bottom. Antibiotics work through precise molecular docking. The drug is shaped to bind to a specific bacterial protein to disrupt the cell's life cycle. Through mutation, bacteria can change the genetic instructions for these target proteins, slightly altering their physical shape. The docking site is no longer recognizable, and the antibiotic passes by without ever engaging its target. Through interception, ejection, and modification, these pathogens have successfully bypassed the specific molecular barriers that human chemical engineering spent decades constructing. If these systems rely on random mutations, how can resistance spread across the entire globe in such a short amount of time? Pathogens use an evolutionary shortcut called horizontal gene transfer. Bacteria carry these defense instructions on plasmids, small, mobile loops of DNA. They connect with other cells to directly trade the blueprints for resistance. This does not require vertical inheritance. They can copy and share these plasmids with entirely different bacterial species. This means a lethal escape pathogen in a hospital setting can acquire a new resistance trait from a completely harmless bacterium in the environment. Multiple resistance genes can be stacked on a single plasmid, allowing a bacterium to acquire multi-drug resistance in a single exchange. Horizontal gene transfer acts as an open-source biological network, allowing pathogens to rapidly crowdsource and deploy defenses in real time. We are trying to match this rapid, decentralized genetic sharing against a human drug development process that is incredibly slow and linear. While the discovery of new resistance genes has exploded, human innovation has stalled. No fundamentally new classes of antibiotics have been approved since 2003. Discovering molecules that can kill bacteria while remaining safe for human biology is chemically difficult and increasingly hard to fund. Because of this stagnation, doctors are often forced to use older, highly toxic last resort drugs like colistin because no other options exist. Even these last resort options are now beginning to fail as pathogens like Klebsiella pneumoniae develop specialized enzymes to neutralize them. We are facing an exponentially accelerating biological threat with a chemical arsenal that is decades out of date. Simply modifying existing chemical formulas is no longer a sustainable long-term strategy against the escape pathogens. Modern research is shifting toward alternative therapeutics that don't rely on traditional chemical pathways. One major avenue is bacteriophage therapy. These are naturally occurring viruses that are evolved specifically to hunt and kill bacteria. Another strategy involves antimicrobial peptides, which bypass molecular docking by physically tearing open the bacterial membrane. While these therapies are promising, most are still in early development in clinical trials, leaving us currently exposed. To survive this crisis, we must abandon the search for a static chemical cure and instead build a medical framework capable of outpacing bacterial adaptation.